In this lecture, we're going to be looking at the chromosomal basis of inheritance. Mendelian inheritance had its physical basis in the behavior of the chromosomes. We know that Mendel's genes have specific locations on the chromosomes, and it's the chromosomes that undergo segregation and independent assortment, so therefore the genes are also going to go with it. The law of segregation says that the two alleles for each gene separate during gamete formation. The law of independent assortment says the alleles of the genes on non-homologous chromosomes assort independently during gamete formation. So for each non-homologous chromosome, it's got a 50-50 chance of which of the gametes it ends up in. We call this the chromosomal theory of inheritance. So there were other people who were working on research. Thomas Morgan was an experimental embryologist in the early 20th century from Columbia University. He was initially skeptical of Mendelism and the chromosome theory. What he studied was the Drosophila melanogaster fruit fly. He used them because they were prolific breeders. There's a new generation every 2 weeks. They only have 4 pairs of chromosomes, 3 autosomes, and 1 sex chromosome. He defined the wild type as the phenotype for a character that you observed most commonly in natural populations. The mutant phenotype was going to represent the traits that were the alternative to the wild type, because these have alleles that were assumed to be mutations from the wild type. He studied eye color in his fruit flies, and what he ended up observing was a correlation between a particular trait and the individual's sex. In his experiment here, you've got the parental generation here, a female with red eyes and a male with white eyes. In the F1 generation, all the offspring had red eyes, just like what we saw with Mendel. In the F2 generation, you had a 3-to-1 ratio of red eyes to white eyes, just like what you saw in Mendel, except that you had all the females having red eyes and only the one of the males. His conclusion was that in the P generation, this was located on the X chromosome. So here you can see the eggs. You've got the gene for the red eyes, and then on the sperm, the gene for the white eyes in the F1 generation. When you looked at the F2 generation here, you had genes for white and red and white for the female, and then just red for the male, none on the Y chromosome. So sex-linked genes are going to exhibit unique patterns of inheritance. The reason for this is in humans and mammals, you get 2 varieties of your sex chromosomes. 2 X's is going to be female, an X and a Y is going to be a male. You're going to inherit 1 chromosome from each parent. So although these chromosomes will somewhat behave like they homologous chromosomes, there's only certain regions that would actually correspond. You have short segments on either end of the Y chromosome that are homologous with the corresponding regions of the X chromosome. So they'll behave like a homologous chromosome without being duplicate copies of all the genes. So in a female, when she produces her gametes, both of them will have X chromosomes. In a male, when he produces the gametes, 50% will have an X, 50% will have a Y. In human, signs of sex begin to appear at about 2 months. Prior to that, the gonad development is generic. Without any further signaling, it would continue on the pathway that develops as a female. In 1990, they identified the Sry gene. It stands for sex-determining region of Y on the Y chromosome. It codes for a protein that regulates other genes that are required for forming a male. So it's required for testes development. On the Y chromosome, there are 78 genes, codes for 25 proteins. Some of them are duplicates, and only about half are expressed in the testes. Sex-linked genes are going to be genes located on either sex chromosome. Y-linked genes are on the Y chromosome. They're passed from fathers to sons. They only go to sons because if you get a Y chromosome, you will be a male. There's very few genes and very few Y-linked disorders just because of the smallness of the Y chromosome. X-linked genes, there's about 1,100 genes on the X chromosome. They get a different pattern of inheritance, since males and females will inherit different numbers of these chromosomes rather than inheriting them the way they do in the autosomal genes. This diagram here, where you can see having the female and the male and the pattern of inheritance, it allows you to compare it where you have a dominant and recessive with recessive, and how the pattern will be different in the males and females. Rather than trying to memorize patterns, the easiest way to be able to do this is to know how to do your Punnett squares. For sex-linked genes, you're going to use either the X or the Y chromosome, whichever the one the gene is on. So here you have the X. This is an X-linked gene. Dominant for the trait would be big N. Recessive for the trait would be small n. There is nothing on the Y chromosome. So what happens is if you inherit a Y chromosome and you have whatever is on the X chromosome, you will manifest it, whether it is dominant or recessive, because you don't have another option to potentially override it. When we look at the inheritance of X-linked genes, X chromosome is going to have several genes that are unrelated to sex, which means they can have disorders that are unrelated to sex as well. The father passes his X-linked alleles to all of their daughters and none of their sons. The father is what is going to determine the sex in the animal. So if you get an X chromosome, you become a female. If you get a Y chromosome, you become a male. The mother passes her X-linked alleles to both sons and daughters. All she has are X chromosomes to pass on. The X-linked recessive traits are only going to be expressed in a female if she's homozygous. So a female would have to inherit it from both parents. Males only have one locus, so any male that receives a recessive allele from their mother will express the trait. So therefore you have more males than females with X-linked disorders. Color blindness is one example of one that's almost always inherited as an X-linked disorder. Duchenne's muscular dystrophy, this is X-linked. It affects 1 in 3,500 males in the U.S. It involves a progressive weakening of the muscles and loss of coordination. These individuals rarely live past their 20s. They're missing a protein called dystrophin. Another example of an X-linked recessive disorder is hemophilia. They're missing one or more of the proteins for blood clotting. So in the female, you're going to inherit 2 X chromosomes. In most of them, they're going to have one inactivated during embryonic development. So the cells are fully prepared to operate with just one X chromosome. The bar body is the inactive X chromosome in each of the cells, and they will in the female. These condense into compact bar bodies. They're reactivated in the ovaries to give rise to the eggs, so all of the female gametes will still get 2 X. Will get an X chromosome. There will be 2 of them at the time when you go to divide them. It's random which chromosome turns into a bar body and independent in each embryonic cell at the time the X inactivation occurs. So a female is a mosaic of the 2 types. You can have some of mom's inactivate, some of dad's inactivate, so you'll actually have a mosaic pattern between mom and dad. After X inactivation, all the mitotic descendants of that cell are going to have the same inactive X. Inactivation is going to involve modification of the DNA and histone proteins bound to it, so it will attach methyl groups to the nitrogenous bases. Your X inactive specific transcript is going to become active only on the chromosome that will become the bar body. That's what makes it become the bar body. So linked genes, these tend to be inherited together because they're located near each other on the same chromosome. So how this affects inheritance is traits that are usually inherited in specific parental combinations. And the reason for this is genes for characters that are near each other on the same chromosome will have a tendency to be inherited together. Genetic recombination is the production of offspring with combination of traits different from either parent. So the recombination of unlinked genes, when you have independent assortment of the chromosome, this will occur. Your parental type is the phenotype of the parent generation. Your recombinance or recombinant type is the new combination of phenotypes in the offspring. So if you're looking at any two genes located on different chromosomes, there's a 50% frequency of recombination. So they cannot be inherited together. It's a 50-50 chance for each of those that you would inherit them in that gamete. The recombination of linked genes crossing over can affect this. So two genes of the same chromosome of the parental type occur with a frequency of greater than 50%. So if the two genes are located on the same chromosome, there's over 50% of the time that they are going to be inherited together. Crossing over is going to account for recombination in linked genes. So while your replicated homologous chromosomes are paired during prophase of meiosis I, there's a set of proteins that orchestrates an exchange of the corresponding segments of one maternal and one paternal chromatid. Here, for example, you can see in meiosis I, you have the dark red and the pink chromosome here that are your homologous chromosomes. A portion of the red and the pink have switched places. So now you have new recombinant chromosomes. So this gives new combinations of alleles, which is more variations for natural selection. So your variations are all for natural selection are going to come from crossing over, making recombinant chromosomes, and random fertilization. All of these increase genetic variations and provide the opportunity for natural selection to work. So what you can see here is you can have these 4 possible different combinations in the gametes rather than having just 2 possible combinations for the gametes. When we go to map genetic information, well, it may sound like a great idea just to draw them all out on a piece of paper and list all the different genes. When you look at the X chromosome having 1,100 genes, and that's only 1.5% of the DNA, when you look at the practicality of writing that out on a map, or even putting it on a computer screen, it's pretty impractical. So instead, what we will do is create maps that are going to look at just specific information. So if you go to look at Google Maps, there is different types of maps you can look at. You can have some that will show terrain, some that will just show streets, some that are going to show a street view. If you put them all on there together, it's going to be a mess and impossible to read. Same thing with the genetic map. So we can make the maps based on the type of information that we're comparing. A genetic map is just the ordered list of the genetic loci located along a particular chromosome. Recombination frequency is the percentage of recombinant offspring. This depends on the distance of the genes between the chromosomes. It makes the assumption that crossing over is random. Alfred Sturtevant proposed this idea that the further apart the two genes are, the higher the probability that a crossover event will occur between them, and a higher recombination frequency. So the closer the genes are to each other, the less likely they will be separated by a crossing over. If they're farther apart, there's a better chance that a crossing over will occur and separate them onto 2 different chromosomes. A linkage map is just a genetic map based on recombination frequencies. With these, the map units are going to be the distance between the genes, and it defines 1 unit as the equivalent to 1% recombination recovery. So despite being on the same chromosome and being physically connected, the genes are genetically unlinked and assort independently. Linkage maps are going to be based strictly on recombination frequency. Cytogenic maps, these locate their genes with respect to chromosomal features, things like staining. What we find with the different types of maps is the linear order of the genes is identical, but the spacing between them is not. Now we're going to look at alterations of chromosome number or structure and genetic disorders. Large-scale chromosomal alterations in humans and mammals often result in spontaneous abortion. Most of these will not survive. Those who do commonly will have developmental disorders. How we get this abnormal chromosome number is a non-disjunction. You have members of a pair of homologous chromosomes that don't move apart properly during meiosis 1, or sister chromatids that fail to separate during meiosis 2. Here you can see if you have a non-disjunction in meiosis 1, over here on the left, one cell is going to get 3, one's only going to get 1. So what you have are half the gametes have an extra chromosome, half are going to be missing some. The other place things can go wrong is in meiosis 2. So they go through meiosis 1, everything's normal. In meiosis 2, you have this non-disjunction. One of the gametes will have an extra, one will be short, and 2 will be normal. Aneuploidy is when the zygote has an abnormal number of a particular chromosome. Monosomy is when you have fertilization involving a gamete that has no copy of a particular chromosome. So it's going to lead to missing a chromosome in the zygote. Most of these are not viable. There's only one that's viable in humans. Trisomy has a chromosome that's present in triplicate in the zygote. There are a few more of those that will survive. Polyploidy is an organism with more than 2 complete chromosome sets in all the somatic cells. This is fairly common in plants, but less common in animals. One of the reasons it works in plants is they have the option for self-pollination. You can find another gamete that will have a similar number of chromosomes. Animals are not able to self-pollinate, so if they have a mismatched number of chromosomes, they're not going to be able to have fertilization occur. When you alter the chromosome structure, this can be a problem. A deletion occurs when you have a fragment that is lost. The deleted fragment can become attached as an extra segment to a sister chromatid. When it does that, we call it a duplication. That's just the deleted fragment on that sister chromatid. We can have an inversion where that chromosomal fragment that comes off will reattach to the original chromosome, but it does it in the reverse orientation. It flips it around. With the translocation, you have the fragment join a non-homologous chromosome. The fragment comes off and decides it's going to just go live on another chromosome. Large deletions in the X chromosome tend to be lethal in the male. They only have one chromosome. There's no chance of really having it turn to a bar body and be hidden. The duplications and translocations tend to be harmful. Some examples of human disorders due to chromosomal alterations. Down syndrome or trisomy 21. This involves 1 in 700 children born in the U.S. There's an extra 21st chromosome. They'll tend to have characteristic facial features, be rather short, have correctable heart defects, developmental delays. They have an increased chance of leukemia. It's about a 20 times greater chance. They will tend to have Alzheimer's show up at a younger age, often in their mid-30s. There's a decreased risk of high blood pressure and atherosclerosis. And there is increased frequency of this with increasing maternal age. Most are due to a non-disjunction during meiosis 1. With all of the chromosomal disorders, we tend to see increased incidence with older mothers. There's a slight increase in incidence with older fathers, but the increase is not nearly as severe as it is with older mothers. Older fathers do tend to have their own separate set of health issues that will come along. Aneuploidy of the sex chromosomes. This appears to have less effect than it does on the autosomes. Your Y chromosome has relatively few genes, and extra X chromosomes can be inactivated as bar bodies in the somatic cells, which will help to absorb any differences. With Klinefelter Syndrome, this is XXY. It's 1 in 500 to 1,000 live male births. They will have male sex organs. The testes are small, they're sterile. The extra X is inactivated. You may see some breast enlargement and female body characteristics. Most will have subnormal intelligence. XYY is sometimes referred to as the super male. It's about 1 in 1,000 males. They have normal sexual development. There's no well-defined syndrome, but they are taller than average. These have been a little bit harder to pinpoint because you don't necessarily know when somebody has a chromosomal abnormality unless they're looking to test for it because something has gone wrong. So XXX This is 1 in 1,000 females. They're healthy, no unusual features other than they're taller than average. They are fertile. They have increased risk for learning disabilities. Turner syndrome, or monosomy X, this is the only known viable monosomy in humans. It's about 1 in 2,500 female births. Phenotypically, they're female, sterile. Their sex organs don't typically mature. Girls that have Turner syndrome can develop secondary sex characteristics with estrogen therapy. They'll tend to have normal intelligence. These are some structurally altered chromosomes. With Klinefelter, this is a specific deletion in chromosome 5. These individuals are severely intellectually disabled, tend to have small heads with unusual facial features, and their cry sounds like a distressed cat. These generally do not live real long. They'll die as an infant or a young child. So chromosomal translocations can be implicated in certain cancers, in particular chronic myelogenous leukemia. This is a reciprocal translocation that occurs during mitosis of the cells that will become white blood cells. They exchange a large portion of chromosome 22 with a small fragment off of the tip of chromosome 9, and this produces what we call the Philadelphia chromosome. The Philadelphia chromosome is just that shortened version of chromosome 22, and it leads to uncontrolled cell cycle progression. Cancer is uncontrolled cell growth. Some of the inheritance patterns are exceptions to standard Mendelian inheritance. If we look at genomic imprinting, you can have a variation in the phenotype depending on whether or not you inherited that allele from the male or female parent. In many cases, it just involves a methyl being added to a cytosine on one of the alleles. So this occurs during gamete formation, and it will result in silencing of a particular allele of certain genes because they're imprinted differently on the sperm and egg. So the zygote expresses only one allele of the imprinted gene inherited from the male or female parent. So it will give you a different pattern depending on whether or not you inherited it from mom or dad. The old imprints erase each generation, so when you go to make your new gametes, you're going to put your own imprint on according to your sex for your gametes. So genomic imprinting is thought to only affect a small fraction of the mammal genome. Most of the known imprinted genes are critical for embryological development. So the example here is in a mouse. If you have the allele inherited from the mother, you get a normal-sized mouse. If it's inherited from the father, you would get a dwarf-sized mouse. So our organelles, some of those can have some genes. They are called extranuclear genes or cytoplasmic genes. They're outside the nucleus, and you'll find these in the mitochondria, the chloroplasts, and other plastids in plants. They contain small circular DNA that's going to have genes on it. These organelles are going to be able to reproduce themselves and transmit the genes to daughter organelles, so they don't display Mendelian inheritance. In most plants and animals, the zygote is going to receive its cytoplasm from the egg, so these are going to be inherited from the maternal side. Most mitochondrial genes make up the protein complexes of the electron transport chain and ATP synthase. So defects in these reduce the amount of ATP a cell can make, which is pretty important. One of the things is your energy levels and how much energy you have to fuel so many other processes in your cells. The muscles and nervous system are susceptible to diseases as a result. One example is myocardial myopathy, which causes weakness, intolerance of exercise, and muscle deterioration. Another example is Leber's hereditary optic neuropathy. This can produce sudden blindness at a relatively young age in people as early as their 20s or 30s. So far, they've identified 4 mutations to cause this disorder that will affect oxidative phosphorylation. These mitochondrial mutations inherited from the mother may at least contribute to some cases of diabetes, heart disease, and several other diseases like Alzheimer's.